Hi there. In this video we're going to take a look at an interesting medium known as Simmons Citrate. Simmons Citrate is kind of pretty looking. It's actually a solid slant type media, and when it's sterile it's green. The green is due to the chemical pH indicator bromethymol blue. It's an interesting name because actually when bromethymol blue is at a neutral pH, it's dark green. It changes to a blue color at pHs above 7.5. So if we get an alkaline condition in here, this should turn blue, and we'll talk about that in just a moment. There are two other chemicals in here we want to talk about. Citrate and ammonium dihydrogen phosphate. Citrate is the organism's only source for carbon. So we all know we're on a planet where we have carbon-based life forms. All cells need carbon in order to grow. All cells also need nitrogen, and the ammonium dihydrogen phosphate is the only source of nitrogen for the cell. So in order to make DNA and protein, we've got to have nitrogen. Citrate gives us carbon. Ammonium dihydrogen phosphate gives us nitrogen. We inoculate the slant like you would inoculate any other slant by starting with a nicely sterile loop. Grab our bacteria, in this case E. coli. We take the loop, put it near the bottom of the slant part of this media, and just drag it carefully, slowly to the top. We've inoculated our slant, flamed the loop. Now this would get a label and would go into our incubator for 24 to 48 hours waiting on a positive result. If it's positive in 24 hours, you're done, but if it's not, you've got to give it another 48 hours. How do we know it's positive, and what do the positive results mean? If the media turns blue, then we know a couple of things have happened. First of all, the organism is growing, so it's using our citrate. It's growing and using the citrate as a carbon source, which means that it has the enzyme citrase. The second thing we know is that it brought in and used the ammonium dihydrogen phosphate, converting that either to ammonia or to ammonium hydroxide. That would create an alkaline condition, which is going to take our bromethymol blue away from neutral, above 7.5, turning it blue. Look at this tube, which stayed green. This would be a citrate negative organism, compared to this tube, which turned blue. This one being blue is very much citrate positive. It means it used the citrate for a carbon source, and the blue tells us that the ammonium dihydrogen phosphate was converted either to ammonia or to ammonium hydroxide. That would have made the interior alkaline, getting us above 7.5 and turning our bromethymol blue actually blue instead of the original green. That's how you interpret Simmons citrate. Thanks.